Hello and welcome to the next instalment of the Oxford Centre for Personalised Medicine Flash interview series, where we talk to individuals playing important roles in personalised medicine in one way or another. Today's guest is Emma Stanton, the Clinical VP of Oxford Nanopore and the first Head of Oxford Nanopore Diagnostics, or OND. Hi Emma, thanks so much for agreeing to chat to me today. So let's kick off with me asking you a little bit more about yourself, your background and your career to date.
Great. Hi, Catherine. Thank you so much for the opportunity to have a conversation with you today. So my name is Emma Stanton and I'm a medical doctor by background. I graduated over 20 years ago from Southampton University, and I always wanted to be a surgeon throughout my medical training. So I initially embarked on my surgical training with a particular interest in upper GI surgery. But as a junior surgeon, you spend a lot of time in A&E, seeing people who are in particular pain. And I was very struck by the number of people that were in pain who presented to A&E that didn't have a surgical cause for that pain that I could fix. And that ultimately led me to switch my medical career from surgeon into becoming a psychiatrist. So I trained at South London and Maudsley as a psychiatrist. And during my psychiatry training, I had the opportunity to take on some non-clinical experiences alongside my clinical training. So I had the opportunity to be a clinical advisor to the chief medical officer, who at the time was Liam Donaldson. And that was during swine flu, so a prior pandemic. And I became very interested in the way health systems worked. And it was that led me to do an MBA, a master's in business administration at Imperial College Business School in London. And I then had the opportunity to do a Harkness fellowship at Harvard in the US. And it was really that international experience that took me out of clinical medicine. I had the opportunity to set up a UK subsidiary of a US managed mental healthcare company called Beacon, and then spent a number of years working for that organization in the US at a time when Obamacare and accountable care organizations are being rolled out across the US. Then more recently, I worked as chief executive for Four Eyes Insight, which was an organization committed to improving operations in the NHS. And then after a period of time, which we may come back to, supporting the British government's response to the COVID pandemic, where I worked in test and trace. I'm really excited to share a little bit more with you about my current role, which brings together a number of those pieces, working clinically, working in government and working in industry, where I'm now working for Oxford Nanopore Technologies, and I'm based here in our headquarters, which is in Oxford Science Park.
Brilliant. So if I say personalised medicine, what does that phrase mean to you?
So as a clinician, personalised medicine sounds like the Holy Grail, because too often we are treating with broad spectrum responses and following guidelines. So I'm excited about when personalised medicine becomes much more accessible. And my current role here at Oxford Nanopore, where I'm the first medical doctor here, and I'm leading the diagnostics team, is really about how we can use the power of sequencing to make personalised medicine a reality. And what I believe about personalised medicine is that it's going to be applicable across a number of different healthcare applications. And what I'm excited about working on with our sequencing technology is it identifying both in the short, medium and long term, where we can use the Nanopore platform to embed it in patient pathways that will really make a difference to patient outcomes for both them and their families. And what I get really excited about isn't just where we are with the technology, but it's the impact of that technology and where we can use it so that doctors with people with health conditions can be empowered to make decisions that will help them get back to work, that will help them get back to doing things that matter to them. And I see personalised medicine really as being the way to unlock that.
So for those of you who don't know, could you actually tell us more about Oxford Nanopore and what it is that the company does? And then what does your role as clinical VP and head of OND involve on a kind of day-to-day basis?
Cool. So Oxford Nanopore Technologies is originally a spin-out company from Oxford University that was founded over 15 years ago. And since it was spun out of the university, it's had almost £1 billion in research funding invested in the platform. And that has enabled unique sensing technology to sequence DNA and RNA in near to real time. And the difference in the sequencing technology that Oxford Nanopore brings is in its level of accuracy, but also in its accessibility. So unlike other sequencing platforms, Oxford Nanopore is on devices like this that hopefully you can see, and the flow cell goes in here, where it's really miniaturized and democratized this approach to sequencing. This doesn't happen today. Almost all sequencing is done in massive, centralized, expensive, high-cost facilities. And what we really stand for at Oxford Nanopore is finding a way to get that sequencing out and accessible to people so that anyone can use it wherever they happen to be in low resource settings, in rural settings. We've even supported astronauts to do sequencing in space. That's really the idea behind the company is to disrupt the existing sequencing market. And perhaps one of the best examples of what this technology can now be used for has been during the pandemic, where a huge proportion of the new variants have actually been both identified and subsequently sequenced, and governments have chosen to use the Nanopore platform to do that. So I think the pandemic for us, and we've been involved at Oxford Nanopore at identifying prior pandemics, but particularly during COVID-19, we've been able to show how this platform can be used in public health laboratories to identify new variants, and that has led to policy and decision making. So coming out of the pandemic, whilst we know there will be a role for ongoing surveillance, my role as head of diagnostics here at Oxford Nanopore is to identify what are the future healthcare applications of this sequencing technology now that we have the accuracy at sufficient level that clinicians and health professionals can use it in a reliable way. Your question about what my role involves on a day-to-day basis is thinking about this problem of how do we use this sequencing technology, which are the areas that it's most applicable to. And just to give you a flavour for some of the applications of this tech in healthcare, we are currently running beta programs for the use of HLA typing for deceased donors to improve outcomes for people where they have transplant organs, so that we know that through using nanopore sequencing, it's a lower cost, it's a faster turnaround time. If you need to do organ donor matching in the middle of the night, for example, you don't need to wait until you've got multiple samples. You can't wait. You often need to do the matching as soon as you have the opportunity. And nanopore sequencing enables you to do that. The beta programs that we're currently working on with partners will enable us to track those improved outcomes, excuse excuse me, so that we can roll this out to wider population. That's one example that we're working on. Another example, just to give you a bit of a sense of the range of applications for the technology, are that we are working with our partners at Guy's and St Thomas's Hospital on when they have patients in intensive care units, they often need support breathing using ventilator, and they're often on support called ECMO. And for those patients who are often extremely unwell, we can use nanopore sequencing to look and see what the underlying cause of infection is. And we do this when the doctors do a ward round in the morning. We can take sputum or other samples to test using nanopore sequencing. And within a range of 4 to 7 hours, that sequencing will identify not only what the underlying infection is, but also what that underlying infection is sensitive to. And the current evaluation that we're doing, which now has over 100 patients that have gone through this workflow using nanopore sequencing, has shown is that in a third of cases that patient who's extremely unwell in an intensive care unit, which costs thousands of pounds a day, is on an incorrect antibiotic. The antibiotic they've been given is broad spectrum and wouldn't be sensitive to the underlying infection that individual has. And so by changing the antibiotic on the same day that we've done that sequencing, we're able to improve that patient's outcome and mean that they can hopefully spend less time on the intensive care unit.
Lovely, thank you. So as someone approaching personalised healthcare from kind of the industrial perspective, do you feel that the challenges that you face are different from those approaching personalised medicine on the clinical or the academic side? And how important do you think it is that the NHS, academic researchers and industrial partners work together to implement personalised medicine?
So part of how Oxford Nanopore has improved its accuracy and developed its technology is by really early on working closely with a community of academic researchers globally. And what that means is that we often do early product releases. We upgrade the technology, the chemistry and the kits fairly regularly and put that out into the academic community to give us feedback, recognizing these are early releases, but each early release leads to a subsequent improvement in the technology. That approach is disruptive. And what that means is that Oxford Nanopore has very close interdependent relationships with academics globally across a very wide range of domains. And we employ about over 800 people in Oxford Nanopore. Many of those people have come from academia. Many have come from Oxford University, but also other universities as well. But we rely on supporting academics to work with us in partnership and publish independent publications showing what our technology is now capable of to be successful. And we want to support our academic colleagues to be successful. But I think it's a fundamentally different relationship that we at Nanopore as an industry partner have with the academic community than I've seen in other industry settings. Another example of it, what I think is a step change in relationships here, is the relationship that we at Oxford Nanopore have with Genomics England, which is an arm's length body from NHS EI, who they are an organisation that we work closely with, and they run a number of programmes around, at the moment, around oncology, around newborn screening, and around improving the diversity of data, and Genomics England has really been set up in a way that will use research and develop research programs so that they can then be rolled out into clinical practice. So whilst we are currently working with Genomics England, particularly in the UK, and perhaps I'm biased towards wanting to do this, but our end goal will be to work more closely with the National Health System in England and indeed other health systems globally. But Genomics England are a key partner for us, improving this technology and working with academics as part of it. So I think there is a lot of overlap across industry, academia, and indeed healthcare, particularly with this technology.
So looking forwards then to the future, where do you see the field of personalised medicine heading in the next 5 to 10 years? And what do you think the biggest challenges are going to be, and how are we going to overcome those hurdles?
It's a great question, Catherine. I care most about the interaction that a clinician has with an individual when they have a health problem. And I care most about developing a technology and a tool that can be used to help both the person and their family with the health problem and the doctor to come to the best decision for them that will improve the outcomes that matter for them. And so I see personalised medicine being a way to unlock that. In terms of some of the challenges that I think, the example that I gave earlier on in the discussion was how we are using sequencing, nanopore sequencing, to identify specific infections in cases that would have otherwise taken days to get the answer to. I think personalised medicine will make the biggest contribution where that time difference matters to patients. So there will be some isolated examples we can use, but we at Oxford Nanopore see sequencing as replacing culture in path labs, and that is not a vision that is shared today by people working in path labs. And so somehow we have to take people on a journey from where they are today to show them that it will be easier, not harder, to adopt new technologies. And I think some of the biggest challenges lie in getting people in path labs to do some things differently and to change some of the things that are currently being done in a centralized fashion to a decentralized fashion. And particularly focusing on the NHS, we have a system that is hugely under pressure with overworked people being asked to do more every day. And somehow we have to find a way where people want to adopt innovative new technologies and see it as being a good thing both for them as well as the people they're trying to treat. Otherwise, those are going to be some of the rate limiting steps. However much we improve the accuracy of our technology, the real challenge is going to be the behavioural change that it will take to get that embedded, not in a pilot, not in an innovation setting, but in a routine clinical care setting. And then there is no doubt in the next 5 to 10 years, the biggest opportunity for personalised medicine lies in oncology. And there is a huge amount of research to do to make that a reality and to prove out the evidence. A statistic that is often shocking is the number of years it takes from knowing something works in a research setting to actually make it happen in a clinical setting. It takes over 17 years for many innovations to reach standardized clinical care. And I know this just from practicing clinically, even when it does reach clinical care, there remains huge variation across the NHS. So someone presenting in the same way in Manchester wouldn't necessarily get treated in the same way in Cornwall, which is fine if those treatments are being tailored to those individuals. But I think we still have a long way to go to make sure that everybody has access to the very best of personalised medicine as we introduce these new approaches.
Well, thank you so much for your time today. It's been really great talking to you.